Okay, so we left off talking about how we get genetic variation. The first one was independent assortment of chromosomes. So this is where we left off last time. So remember that each pair, when they line up down the center of the cell for meiosis, can line up in one of two different ways. And so therefore it's 2 to the nth power to figure out how many different combinations, where n is 23 for humans, because that's our haploid number. So we learned that there are more than 8 million combinations just in the way you have to imagine, the chromosomes lining up. We have 23 pairs of chromosomes down the center. If my hands are the 2 chromosomes, and each chromosome can line up in one of 2 different ways. So this one can line up one of 2 different ways, and then all the other ones can line up like this or like this. And so therefore you get lots, just more than 8 million combinations just with the way that they line up, meaning that you have, just with the way that they line up, you have 1 in 8 million chances of ever making the same egg cell twice in your lifetime if you're a female, or sperm cell if you're a male. All right, so let's turn the page. Alrighty, let's see here. So let's look at that. All right, we're going to do some color coding here, and I'm going to say here that we have 2 pairs of chromosomes. So notice here it shows you a key here, but there is, it looks, both of them, the boxes look the same color because it's hard to tell there. Let's pick a couple of different colors here. I'll pick pink, so pink for maternal. All right, chromosomes. Actually, my pink has seen better days. Let's do, let's change it to purple. Sorry, guys. Purple. I just don't want to run out of ink while I'm going over here. So maternal, and let's do blue for paternal. All right, so remember, one set comes from mom, one set comes from dad. So I'm going to go ahead and put the blue. So this is 1/2 of the chromosome. This is the other half. 1/2 of this one, other half. And so this is metaphase 1, where they're lining up with their pairs. I know these are pairs because they're one large pair and one small pair. All right, and so that's what we have here. All right, so then looking at this, let's assign some genes. All right, so let's say the person, I'm just going to do a different color here. Well, I'll just do in red. All right, so over here, let's say we have 2 different genes. Let's say they're big D, big R, little r. All right, and so let's assign what these things mean. It doesn't really matter what it means, but let's say a big D is having more than 5 fingers. All right, that's called polydactyly. That's actually a dominant trait. And little d is having 5 fingers. And let's say a big R is having sickle cell disease. It's a disease that, remember, changes the shape of the red blood cell. Sickle cell disease and no sickle cell disease. All right, so that gives some meaning to our letters. So therefore, if they're big D, big R, little r, does this person, what's true about them? So for the 5-finger trait, they have, what, more than 5 fingers, right? And for the R trait, they have sickle cell disease. Oh, and you know what? I just realized I wrote this wrong. This should be a little r. This should be a big R. Sorry, I got carried away there. All right, and so that's what we have there. So no sickle cell disease is dominant. Sickle cell disease is recessive. So this person has more than 5 fingers and does not have sickle cell disease. So let's write this on there. So what does this mean in terms of their chromosomes? Let's say the DNA that decides how many fingers you have is on the big chromosome. So let's say it's, each one of these is made of a piece of DNA. So right here on the chromosome, I'm going to write it just above it. They each have this is the big D chromosome because they're big D, little d. So one chromosome has a big D, and the other one, since the 2 chromatids are exactly the same, that's exactly the replicated part of the chromosome. That's why this chromosome has two big Ds because of the two sister chromatids are exactly the same DNA. And then the second chromosome has little Ds on it. All right, and let's say the small chromosome has the R's. So the small chromosome has the big R's and the little R. All right, and so, excuse me. So when meiosis one occurs, the pairs separate to opposite sides of the cell. So therefore, we have both blue, large blue, and small blue chromosome goes this way. And going back to the purple, large purple, small purple goes this way, and they end up in different cells. So the pairs separate, and so we have the big D chromosome over here and the big R chromosome over here, little d and little r. All right, now what happens, that's meiosis one. So what happens in meiosis two is the sister chromatids separate. So going back to color coding here, this half is going to go to this side, this is going to go to this side. These two are going to end up in these two cells. All right, and then same thing's going to happen here. This chromatid and this chromatid are going to go over here. So I'm going to color those purple, and these two are going to go over here. So therefore, these are, now what are my combinations? It's the genes that are important here. So I'm going to have one big D, one big R, big D, big R, little d, little r, little d, little r. So therefore, I have two combinations here. As a person of this genotype, having these genes, they can, if their chromosomes line up this way, they can, when they make their egg or sperm cells, they can make one like this or one like this. All right, so they can pass on the greater than 5 fingers and no sickle cell disease, or 5 fingers and sickle cell disease. All right. So then, but then we have possibility 2, because remember, independent assortment means the chromosomes can line up in one of 2 different ways. And so, what happens is when you're looking at 2 pairs here, we're going to flip 1 pair. All right, so I'm going to keep the top pair the same. So again, I'm going to color code here. So this is going to be the blue paternal right here, and this is going to be the maternal. And let's put the genes on there. So that part's going to be the same. So they're going to have big D chromosome, little d chromosomes. All right, so then let's flip-flop these two. These two don't have to, all the paternal chromosomes don't have to be on the left. So they can switch. So let's make the maternal one, let's flip-flop these guys. The maternal one on the left and the paternal one on the right. So that means that you have the little r chromosome over here and the big R chromosome over here. So now what happens in meiosis one, all right, so the pairs separate. So you're going to have this blue chromosome right here, over here, and this purple one over here. And then this purple one over here, and this blue one over here. So therefore, what genes do they have? So that blue one has the big D's, the purple has the little R's, purple has little D's, blue has the big R's. All right, little one. So now in meiosis 2, what separates the chromatids? And so therefore, you're going to have big blue over here, big blue over here, little purple right here, little purple right here. All right, then on this side here, you're going to have the purple chromatid, so a bit too big. This purple chromatid here, this purple over here, and then these guys separate, this one to this side, this one to this side, and so then you have 2 small blue ones. So let's assign the genes on here. So now you have the big D chromosome with the little r, big D with little r, and you can have little d with big R, little d with big R. All right, and so therefore now we have 2 different combinations, combination 3 and 4. So before, when they lined up, you couldn't get a big D and a little r, but now you can. And so therefore, those are your 4 different combinations that you can pass on to your kids, just if you're these genotypes, just by the way that they line up. We're not going to switch because if you switch these again and keep these the same, you're going to end up with the same as over here. All right, so when you're looking at just 2 chromosomes, it only makes sense to flip one of them, or else you're going to end up with the same combinations. All right, so that now let's take into account crossing over. So going down here, crossing over produces what we call recombinant chromosomes. That means that they are recombined. All right, so it combines genes inherited from each parent. Crossing over begins very early in prophase 1, so we talked about that. The very beginning of meiosis, as homologous chromosomes pair up gene by gene. So they come and pair up, and then they do that crossing over. So the homologous portions of 2 non-sister chromatids trade places. That means, so remember, this is one chromosome, one chromatid, and one chromatid. So non-sister, all right, this one chromatid, non-sister would be like this and this, all right? So from 2 different chromosomes, and they have to be of a pair, of a homologous pair. So they have to be matching. So crossing over contributes to genetic variation by combining DNA from 2 parents into a single chromosome. So let's go through this example here about that. All right, so we're going to now look at, we're going to do the same kind of color coding here. So we're going to start out with just 1 pair of chromosomes. In this case here, we're going to throw in and put 3 different genes on this 1 pair of chromosomes. And for simplistic sake, we'll keep the letters the same as last time. But I'm going to do here, I'm going to do blue again and purple. So here's 1 pair. All right, and so here are your non-sister chromatids from the different pairs. All right, and let's say the DNA on this, so let's do DNA here. Let's say the DNA, here's the DNA making up one of these. All right, so what that means, since they're homologous, is that they both have similar DNA in terms of the genes. So if this is the DNA, let's say the person is big D, big N, little n, big R, little r. So now I'm doing 3 different genes. And let's say the big D gene is right here. Let's say the big N is right here, and the R gene is right here. All right, so if you're big D, big N, little n, big R, little r. All right, so what we're going to say is if this is your 2 pieces of DNA, so this is 1, and this is like your chromatin from here to here, this is your gene, you're going to have a little d, little n, and little r. So this is your maternal, and this is your paternal piece of DNA that you got from your mom and dad. And then, but that's what I mean by it. They're homologous, meaning the same types of genes on them. You can just have different versions of the gene. So then when it gets ready to go through meiosis, remember the DNA replicates and you get replicated chromosomes like this, and it condenses and so on. So going back over here, let's put right here that this is this is the big D chromosome, big N and big R, and this one I'm not going to put it, so each side of the chromatids would be the same, have those same genes on it because they're replicated, and this chromosome is going to have the little d, the little n, and the little r. Okay, and so so that's what that looks like. This is the beginning. I'll put this here, the beginning cell. All right, and this is your genotype. And so, so now when it goes through meiosis, if we take into account crossing over, so now what we have here is the blue chromosome right here, and this part here they're going to cross over. So I'm going to put here the purple on here, and then they're going to cross over at this point here. So they're just going to cross over the D genes, all right, where the D genes are located. And so then here's your purple chromosome, all right, and then they're going to cross over down here where the R chromosomes are located. All right, so then they're still crossed over in metaphase. So you're going to have your blue kind of chromosome here, but the ends of the blue one are going to be purple. I'll put the little, or whatever color you're using, and then here's your purple chromosome, And the purple chromosomes end, it's kind of peeking out here, is about right here, are blue. So they're going to literally cross over and swap pieces of DNA. All right, and so therefore, what it's going to look like after these guys separate, and this is again Meiosis One. So let's go ahead and draw. So these chromosomes here are the outside chromatids are still going to have the D, the big N, and the big R, and the little d, the little n, and the little r. And the same thing here, big D, big N right here, and big R. All right, and this one's going to have the little r, the little n, and the little d. Now I want to show you how that makes a difference when they separate. So now when they separate, you have this chromosome, so the blue one's going to go over here. This is not affected, this side is not affected by crossing over, but this side is. And so when these guys separate, this part right here is going to be the same, but the end is going to have the purple part. All right, so the purple part there. So what does that mean in terms of genes? It means that this one is still going to have the big N, or big D, big N, and big R. But instead of this one having the big D, it is now going to have the little d because it's swapped. But then that didn't change the big N. And then this one down here swapped here. All right, so therefore it's going to have, instead of a big R, it's going to have a little r, in which I just realized I was supposed to make that end purple too. So I'm going to make these ends there, and that's going to have a little r. All right, so now, because they did double crossing over here. All right, so then when we look at this one over here, let's do this one as the purple, but again, this side is not affected by the crossing over, but this side is. Both ends, so they're going to draw it purple, but both ends now are going to be blue because they switched spots with these two ends. And so now the genes, this side isn't affected. It's going to have all the recessive still, but this side is going to have the big D, the N is not affected, and it's going to have the big R. So now what happens is when meiosis 2, let's call this meiosis 2, separates, remember that's the chromatids, so you're going to have a complete blue one, the side here, complete blue, with the big D, the big N, and the big R on it. But then this chromatid is going to be blue with the purple ends. So these are little d, big N, little r. This one, this chromatid is going to go to this cell. It's going to be purple and with the blue ends. And the combination of genes it's going to have is the big D, the little n, and the big R. And then this one is going to be all purple. It wasn't affected by crossing over at all. And so therefore, we'll still have the little d, the little n, and the little r. So now you see here, when we're looking at this, that these guys are recombinant chromosomes. Without crossing over, there was the only two possibilities with this that you could have given to your kids is all dominant or all recessive. Now you have a new combination that you can pass on to your kids as a result of crossing over. And so crossing over occurs, this is just showing you one pair of chromosomes with 3 different genes on it. We can have hundreds of different genes on a chromosome, and we have 23 pairs of chromosomes. And so not only, remember, you have the way that they line up with independent assortment, remember, gives you more than 8 million combinations, but now you throw in crossing over, which is totally random and occurs, all the time during meiosis. It gives you new combinations. that really ups the chances, lowers the chances that you will ever make the same egg or sperm cell twice in your lifetime. Which is why you can have family members, you can have parents that have 10 kids, and none of those 10 kids could look alike or even be anything alike. And this is why, even though you're literally half the DNA from your mom, half the DNA from your dad, it has to do with the independent assortment, how they line up in this crossing over. In addition, the third thing is random fertilization. It adds to genetic variation because any sperm can fuse with any ovum or unfertilized egg. The fusion of gametes produces a zygote with any of about 64 trillion diploid combinations. And so this is what we call the rule of multiplication, where the chances that a mom, all right, so there's a 1 in 8 million possible combinations in the egg, just based upon that stat from independent assortment. So 1 in 8 million combinations that mom is ever going to make her the same egg cell again, and then 1 in 8 million possible combinations that dad would ever make that same sperm cell ever again in his lifetime. But then to get the exact same kid, mom would have to make that exact same sperm cell twice. All right. So when she made that, egg cell, twice. So let's say they have a kid. All right, so they have little Johnny, and then to get a second little Johnny, what would need to happen is mom would have to make that same egg cell, and dad would have to make that same sperm cell, and then that exact same sperm cell would have to fertilize that exact same egg cell. And so therefore, to figure out this stat where I get 64 trillion is you take the individual probabilities and multiply them together. So 1 in 8 million chance that mom's going to make the same egg cell twice, 1 in 8 million chance that dad will ever make the same sperm cell twice. The chance that those two individual events are going to occur together, you take the individual probabilities and you multiply them. So you take 1 in 8 million and 1 in 8 million, multiply them, and you get 1 out of 64 trillion. So highly unlikely that you'll ever have the same child born by the same parents again. And this is not even taking into account what? I didn't even take into account crossing over. So then you throw in crossing over, and it's way, way more than 1 in 64 trillion. So that's what this is. Crossing over adds even more variation. All right, so therefore each zygote and therefore each person has their own unique genetic identity because of independent assortment, crossing over, and random fertilization. So this is really important evolutionarily speaking. Natural selection results in accumulation of genetic variations favored by the environment. So it's good that we all have different combinations of genes, even siblings coming from the same parent, in the hopes that somebody has combinations of genes in the population that help the population to survive, even if the environment changes. So sexual reproduction contributes to the genetic variation of the population by these different things that we just talked about, and remember that all of these different genetic variations ultimately results from mutations. Why there's a big D and a little d is because somewhere along the line, everybody was big D, and then somebody's DNA got mutated, and it wasn't harmful, and maybe if it was helpful, and then it got passed on to their kids, and then their kids, and so on, and over thousands and thousands of years, we get a new trait in the environment or in the population. So reproduction processes that increase genetic variation are evolutionarily conserved because they're good. If you increase reproduction that increases genetic variation, that's a good thing because it ensures more likely the survival of the species and of the population. All right, and so therefore very, very important. And that is the end of chapter 10.